Okay, last time I talked to you partly about protein translation. One part was left over because I could not cover on that day. Today I will cover that part. That will be the rest of the part. See, protein once synthesized, as I told you, there are two types of ribosomes. One is free ribosomes, one are bound to endoplasmic reticulum. So there are two types of ribosomes. I told you, free ribosomes and the bound ribosomes. Free ribosomes synthesize basically protein which are required for intracellular requirements, including those required for the membranes. But those which are synthesized by the endoplasmic reticulum are usually transported or exported. So they are processed through the endoplasmic reticulum, Golgi apparatus, and they are transported through the excretory vesicles. So all the proteins whichever have been synthesized in the native form are not functional. They require some modifications, and these modifications are very much important for their optimal function, be it conformation, be it biological activity, be it for binding, be it for affinity, be it for subunit aggregation. Whatever way you can say, the many biochemical processes are going on, and that requires modification, alteration, and at one point of time the enzymes will also be destroyed within the cell and extracellularly also. So there must be mechanisms for generation of synthesis at the time of need and repression of protein synthesis when it is no longer needed, or whether when the cell is suffering from energy crisis. So in energy starved cell, usually synthetic mechanisms are kept involved because protein synthesis is an energy intensive procedure. I told you earlier that for one peptide bond, 4 high energy compounds are required. So post-translational modifications, you have to memorize, you have to remember there is no way out, because this will come as a question, maybe as a short note, maybe asked during the viva, with at least one example. So you have to see, if you want to talk, you talk a little less so that others are not disturbed. I can't make you to stop talking. The way you want to behave in the class, you behave. I don't bother, but do not try to disturb others. That disturbs me also indirectly. So try to remember that I told you prokaryotic basically polycystic, because there are many genes are transcribed together and they are also processed post-translational modification, if at all they are done not post-translationally, they post-translationally modified. And after the protein synthesis has been complete, they are cleaved. So all the individual proteins are released. But in case of eukaryotic, that is basically a complex protein that requires some modification, because this protein synthesis occurs in the cytoplasm as well as in the mitochondria, unlike prokaryotes. So prokaryote synthesis is polycystic. I am repeating for the third time. And eukaryotic is monocystic, and they are synthesized as a precursor molecule. And as I told you that they require modification because of the conformation, because of the binding capacity, because of the affinity, subcellular. As you know, hemoglobin is having 4 units. So that 4 units, if you destroy, the hemoglobin will no longer function. Same thing is IgM. 5 units of IgG molecules are joined together by a joining cell. So without that form, the proteins will not function properly. So one good example is insulin, and you have to, even if you do DM in endocrinology, this will be the first step during any diabetes discussion. There is no way out. The introduction slide will be on this part, that is insulin is synthesized as a pre-pro-insulin, and that is containing 110 amino acids, and that is broken down to pro-insulin and finally to insulin and C-peptide, and both insulin and C-peptide are produced in equimolar concentration. Indirectly, we can estimate the endogenous synthesis. Somebody might be taking insulin exogenously, so that will lower down blood sugar. Somebody might be producing insulin within the body. Whether the person's blood sugar level at a given point of time is because of the endogenous insulin or exogenous insulin, that we have to determine by measuring the C-peptide. Because we will not be able to do this thing. This will be medically important. I am telling you, many patients will come for medical fitness study. Their blood glucose is a mandatory test, because if the patient is diabetic at times, although I am not supposed to tell this thing here, but I am you being a medical graduate, and you will be required to issue a medical certificate. So they will be declared medically fit, they don't have a diabetes, but they are taking insulin. Now if you have a little bit of doubt on that, estimate C-peptide. The C-peptide will be conspicuously absent in those who are taking insulin by injection, not by those who are producing insulin by themselves. And insulin, they are having two chains. I am telling you, A chain and B chain, the total amino acids will be repeatedly asked in the examination. There are 51 intra-chain disulfide bond and inter-chain disulfide bonds. Where these are located, that you must remember. This is the structure of pre-pro-insulin. That is the highly complicated structure. It is having a single peptide. You can see there is a single peptide here. I am just reviewing the single peptide. And this is the common structure which you will see in the book. See, insulin is secreted as a single peptide, as I told you. Now, this single peptide is folding on itself. It is folding on itself within the endoplasmic reticulum. Now, because of the folding, now this chain is formed. You see, there is a connecting peptide. And this connecting peptide is released by the trypsin-like enzymes, which are shown in red, and at both the ends, we are having carboxypeptidases. So it will release basically the connecting peptide, and finally the A chain and B chain will be produced. So as I told you earlier, one molecule of insulin will yield one molecule of C-peptide. So that equimolar concentration will be there, and that is how we will determine whether it is endogenous or exogenous insulin. So this structure, you remember that A chain is having 21 and B chain is having 30. The A chain inter-chain disulfide linkage, that is a primary structure that must come as a short note also. That is between this is 6 and 11, and this is 21, and this is 1921 to 1920, and this is another disulfide bond. But Regina, Jachana, Regina, this is another important thing, is angiotensin. You will be using medications for blood sugar, blood hypertension. So these drugs will be used. So angiotensin, role of renin-angiotensin in maintaining blood pressure is very important. And angiotensin is basically a decapeptide, and it can be produced in three different things through a proteolytic cleavage, 1 to 7, 1 to 8, and 2 to 8. And angiotensin is a precursor for renin, and all the different forms of angiotensin forms of angiotensin are produced by the ACE1 or ACE2 inhibitor. ACE inhibitors you will be using pharmacologically, and for many patients you will be required to give the ACE inhibitor drugs also. This is another example. This you all know. Only thing is that you have to remember this thing and you have to write during the examination. See, zymogen to zymogen. All enzymes, digestive enzymes, are produced in the form of zymogens, and they are activated like pepsinogen. Pepsinogen is converted into pepsin to the ACE cell. That is again a precursor for protein. Similarly, trypsinogen to trypsin. See, 373 amino acids has been converted to 47 amino acids. This is the active pepsin. See, chymotrypsinogen to chymotrypsin, pro-carboxypeptidase, pro-amylopeptidase to amino proteolytic cleavage. Remember, there are two different types of things. One is proteolytic cleavage, one is covalent modification. Here it is a covalent modification. Another covalent modification I am telling you. Now, this list you have to remember. Proteolytic cleavage and covalent modification, these are the two basic steps. So, initiate a methionine removal. As I told you, that first step of protein synthesis is the methionine in case of eukaryote and formyl methionine in case of prokaryote. So, this initiate amino acid needs to be removed. since it is at the amino end, so amino peptidase will be there. So methionine aminopeptidase will clip the basically methionine to remove the initiator proton. Acetylation. Acetylation of a group of protein, and that is required for the activation, particularly histone proteins, for activation and repression of gene expression. It will be required. So regulation of gene expression, I think this has been told, or it will be told. The chromosomal assembly, phosphorylation. You know phosphorylation, dephosphorylation cascade during the glycogen synthesis, that is glycogenesis and glycogenolysis. One is active form, one is inactive form. Phosphorylase, which is basically catalyzing the glycogenolysis, is the phosphorylated form. In contrast, glycogen synthesis, when it is phosphorylated, it is inactive form. So methylation, again methylation protein is arginine lysine, and they regulate the protein-protein interaction. As you go higher up, you will understand the impact of all those things, because these are also used for research purposes. Ubiquitination. This ubiquitination marking that a protein is almost destined for destruction. So gamma carboxylation, vitamin K dependent factor require gamma carboxylation for proper clotting, and glycosylation during your carbohydrate. You have read that carbohydrates are linked either by O-glycosidic linkage or N-glycosidic linkage. So glycosylation, addition of oligosaccharide chain or glycans to the proteins, that causes protein stability, folding, cell addition, signaling, everything. The disulfide bond, I told you, insulin, translation, addition of phosphatic group, and subunit aggregation, as I told you, hemoglobin and immunoglobulin, and also protein folding by chaperones. There will be separate class on chaperones on protein folding. So this is the main difference of prokaryotic and eukaryotic. You will get it from the book. You need not worry that much. Only you must be able to tell at least what is the synthetic machinery. And a prokaryotic, this is Sine-Dalgarno sequence, and this is Koja-Consensus sequence. This is cap dependent and cap independent, and elongation factor is different, termination factors are different. So this you will get it from the book. This you remember. The prokaryotic, the efficiency, translation efficiency is 15 to 20 amino acids per second they can attach. But in contrast, eukaryotic cells only about 2 amino acids per second, because this is very crucial, because if you insert a wrong amino acid, that is going to cause if it is an active site of a protein, if it is a functional protein, then the function of that particular protein might be lost, particularly enzymes. Next. So inhibitors of proteins, these basically are used for research purposes, for understanding. Like puromycin, cyclohexamide, these are not, you will not be using. So these are required for understanding the basic process in the laboratory. They are given in the culture media, or they are given in the culture, this laboratory where the media, where the bacteria are grown. So they are grown, they are allowed, they are given in that the puromycin and cyclohexamide, and they want to see whether the protein is being expressed or not. So basically what does this puromycin does? It's a tyrosine RNA. So it basically blocks the protein synthesis, and cyclohexamide inhibits the peptidyl transferase, which is present in the larger subunit. Similarly, lysine is a toxic molecule which is present in the plant cells, and that inhibits 20S RNA. This you have to remember. This might come as an MCQ question also. This is which one is inhibited by this lysine or puromycin or cyclohexamide. Diphtheria toxin is important for human being, but because of the advent of DPT vaccine, practically you will not be able to see diphtheria cases in your clinical experiences. When it was not known, at that time it causes inactivation of elongation factor 2. So that used to inhibit protein synthesis. And Pseudomonas aeruginosa exoticin A, This you will see in your clinical practice that inhibits the inactivates elongation factor 2, so that also partially decreases or that protein translation. So mitochondrial DNA, I just briefly cover about this thing. The mitochondria is also having a DNA, and their protein synthetic machinery is totally different, and they mainly use or synthesize the proteins which are required for the electron transport chain or speed chain. See, mitochondria we inherit from our mother. We all of us inherit mitochondria from our mother, not from our father. Both paternal mitochondria is present also in the zygote, but unfortunately these are destroyed by there is a special autophagosome which basically destroys mitochondria. So that destroys the paternal mitochondria, but not the maternal mitochondria, and all the diseases which are present, mitochondrial DNA-based diseases, are inherited from the mother exclusively. So you see the unaffected father, unaffected mother. So once the mother is having that disease, that mitochondrial disease, all the offspring will be having that disease. In contrast, if the father is having all the mitochondrial diseases, since paternal mitochondria is destroyed in the zygote, none of them will be affected. You must remember that thing, and this is having also a very good impact in forensic science, because once somebody might be adopting a child, you don't know whether that mother is the biological mother of that child or not. For that, if the mitochondria is analyzed, so mitochondrial DNA sequence of the mother, that if you get it, so he is the actual mother. So in case of disputed parentage, in case of child custody, in case of this, you might across in a very rare situation, but try to remember this thing: mitochondrial inheritance is from the mother and not from the father. So these are the diseases. These are very rare diseases. By chance, in that baby, if this type of diseases are present, and if he is not a biological mother, so this can be ruled out that she is an adopted child. She is at least not the mother's foster mother. Okay. So there are many diseases. Like one important I am telling you, that today diabetes is very common. Little bit. You may be able to detect diabetes at an early age. So in that case, you try to understand whether it is a maternally inherited diabetes mellitus and deafness. If by chance the baby unfortunately develops diabetes and deafness at a very early age, particularly those who are opting for pediatric, they must be very much vigilant about this type of abnormalities. I am telling you, all these genetic disorders, whatever you have read or you will be reading, are indeed all rare diseases. It's true, but we are not clinically that sound also. We cannot diagnose. That is another part. So if your mind knows, your eyes will be able to see it. If you don't know about the disease, you will not be able to diagnose. Like today I will also discuss some of the diseases which you might come across once in a lifetime, provided you know that fact. And it will click in the mind that, yes, I read that book. So in case you get a diabetes, in early childhood, check deafness also. And if that is present, that means this child is having a very bad prognosis. It is not that curable because it's a genetic disease. Okay, so a mitochondrial DNA is having 37 genes, out of which 13 is coding for electron transport chain and 24 is non-coding. Remember, all of you know about inheritances, that is Mendelian inheritances. Mitochondrial inheritance belong to non-Mendelian inheritances. A class of diseases which I read 10 years after post-graduation, that there are one kind of inheritance that is called non-Mendelian inheritance, and I am thinking of delivering a lecture at the national level in this year in our biochemistry conference, annual conference at Coimbatore. Because these many students may not be knowing, many persons may not be aware of non-Mendelian inheritances. So there are 100 copies of mitochondrial DNA and they are supposed to also get mutations. So since they are randomly distributed, so there is a chance that a cell might have a normal mitochondria or a mutated mitochondria, and that is what is called heteroplasty. And because of the accumulation of mutation over a period of time, aging or age-related diseases occur also because of the mitochondrial mutation. Next. So this mitochondria is having two strands, one is A strand, one is L strand. So basically they are having encoding genes at 12, and there are ribosomal subunits at 12 base and 15s. So these are the mitochondrial gene details I am telling you. Just to remember, this slide I showed you earlier also, that mitochondrial codon is not same as cytoplasmic codon, that is mRNA codon present in the cytoplasm. So there are differences and you must remember. This might again come as a MCQ question. So mitochondrial protein synthesis, mitochondria is also having a ribosome, but it is having a 55S and it is having two subunits, 39S and 28S, and their RNA and protein compositions are also different. So I am telling you the enzymes, the mitochondria synthesize proteins which are required for electron transport chain, and these are all 13 in number, 40 polypeptide chain. And mitochondrial DNA is inherited from the mother, and I explained that this is important for genetic diseases and for study of forensic medicine. The mitochondrial genome is having a 16,559 base pair in contrast to human genome, which is having 3.2 to 3.3 billion base pairs. So there are mitochondrial DNA is also having the same steps: initiation, elongation and termination. Only some differences are there. There are one pre-initiation complex, here it is a two pre-initiation complex, mitochondrial pre-initiation complex 1 and 2. An elongation factor is there, is similar to prokaryote, and basically that we have got the mitochondrial DNA from prokaryotes is very much evident evolutionarily because of the protein synthetic mechanism which uses almost the same factors. Like here it is EFTU. EFTU is also present in the prokaryote. EFTS is also present in the prokaryote. So they are also living that they are the same places, that P site, A site and exit site. Only a little bit different because all the factors are not same, like here termination factor is different. So at the end of synthesis, same termination factor will come, it will release the peptide and the peptide will remain within the mitochondria. So these are mitochondrial translation process, you read it a little bit. Nothing more, only thing is that you have to remember all the differences. This is, you just remember somehow that we have inherited mitochondria from prokaryotes, will be clear from here. The prokaryotes, it is circular, it is circular. It is 70S, 70S, 22, TRNA, 22. So initiating amino acid formal method, A formal method. Effect of tetracycline, here it is not affected, here it is inhibited, it is inhibited. Why? Tetracycline is mainly, we take it to kill bacteria. It's a bacteriostatic drug, not bactericidal. It stops the bacterial growth. So why it should stop the mitochondrial DNA synthesis? Because it is having a almost translational machinery, almost similar, if not identical. Of course, not identical. It's similar to prokaryotes. Okay. With this, we conclude now. Next part. This is the today's class, because I am running already 35 minutes late. So this is basically today's class. I'll try to cover as much as I can. Otherwise. So DNA, as you have been told about during replication, that DNA is also, undergoes repair at the time of replication, so that the exact copy is produced. By chance, it happens, it does happen. There is no perfect system. It does happen that DNA polymerase, whatever we are having 5 units. Remember, bacteria are having DNA polymerase 1, 2, 3, but we are having alpha, beta, gamma, delta, epsilon, eukaryotes basically, and they are having different functions. I will show in a little while. After this baby is born, we are exposed to environment, and the entire environment is hostile. There are different types of radiations, ultraviolet radiations, radioactive materials, chemical carcinogens, particularly those who are exposed. Suppose somebody has taken a job in a factory in which they are supposed to deal with the chemicals. So these chemicals act also as a carcinogen, and all these causes damage to the DNA. Body has evolved mechanism over crores of years of evolution to somehow get rid of those damages. But despite all these things, there will be a failure, and then of course that proper rectification will not be done. It might lead to DNA damage and mutation also. So elaborate mechanisms are there. There are a good number of proteins which are beyond the scope of today's or maybe one day's discussion. So many proteins are involved in recognizing the defects, in repairing those defects. These are really innumerable. Many things are known. I am telling you only a very few. So remember this thing. Don't try to get confused. E. coli, this is bacteria. They are having DNA polymerase 1, 2, and 3. But eukaryotes, we are having these. There are 5 different types of DNA polymerases, and since we are concerned with today's DNA repair, remember DNA polymerase 2 is concerned with DNA proofreading and repair. In eukaryotic system, DNA beta, all beta is responsible for DNA repair. But while the leading strand and the lagging strand are being synthesized, they are also having a proofreading capacity. So they also perform this delta, alpha, and epsilon. They are also concerned with some form of DNA repair, as we will understand in a little while. So gamma is basically involved with the mitochondrial DNA synthesis. So error rate, although having a very good replication copying capacity, but in bacteria, there is one mistake for 1 million to 1 billion nucleotides. In contrast, in humans, you see the error rate is one mistake per 100 to 1000 nucleotides. So error rate is quite high in case of eukaryotes. So that is the reason why the synthetic machinery is not that fast as in prokaryotes. So end result, what happens in bacteria? If there is a mutation, bacteria will develop antibody resistance, and bacteria will try to do that thing, because we are trying to kill the bacteria or prokaryotes. They are trying to also survive. So they evolve mechanisms through mutations to survive. And in our system also, we also develop, like one classical example is thalassemia. Because malaria, this I will be discussing maybe tomorrow. See, all the hemoglobinopathies are the basic outcome of long exposure to malaria parasites. The malaria parasites grow on the RBC. So our system has evolved a mechanism so that it reduces the lifespan of RBC. Malaria parasite will not be able to grow. And that is the reason why thalassemia is little bit common in this area, because this area was exposed to mosquitoes even now. Mosquito, particularly Calcutta and other suburbs area. So there is also malaria incidence. So long this malaria will be there, this genetic disease will not go, because body has evolved the genetic mechanism. They have mutated their own gene, the hemoglobin gene, such that the RBC lifespan will be decreased, so that they will not allow parasite to grow. So that is, again I am telling you, so in humans, if there is a mutation, there are two types of cells. One is somatic cells, that is within the body, and one is germline cell, which is present in the gonad. If the somatic cell undergoes mutation, it is likely to cause unregulated cell proliferation, maybe causing a tumor, and if it is too much, then it will cause cancer. If it is a germline mutation, that will be propagated to the next generation, to the gametes. So gonads are to be protected at any cost, be it in male or be it in female, during the entire reproductive life from any kind of exposure which is likely to cause damage to the gonads because of the spermatogenesis or oogenesis, because that defect will be permanent and it will be carried forward to the next generation. So that is called a germline mutation. This is inherited cancer from parents. Sometimes we get diseases from parents, and that is a germline because of germline disease or mutation. So too much of good things of anything is very, very bad. So body has also developed some imperfectness during synthetic mechanisms for which the exact copying, exact translation, exact replication doesn't occur to give space for evolution to occur. So they develop mechanisms, and that is the reason what we have, or eukaryotic cells have developed the DNA synthetic mechanisms are not same as that what is present in the prokaryotes. So evolution to continue, some kind of imperfectness is required. And as a rule, no person in the world is a perfect person. No person. So to finding perfectness in anybody, in any way, is basically a wrong approach. So for survival of propagation, there is an elaborate mechanism to maintain the integrity of the DNA, both at the nucleotide sequence level and at the chromosomal level, because chromosome basically, when the DNA is interacted with the proteins, they form the histone and non-histone proteins, they form the chromosome and they regulate the gene expression. So checkpoint controls are there during the cell cycle. In a short while you will see, to halt progression if the DNA damage is found at any of the stages. It will not allow the next stage to progress. Ultimately, either the cell will undergo arrest until the repair is repaired, or it will, if not, cannot be done, the repair cannot be done, it might lead to apoptosis, or it might lead to senescence also. Senescence means it will be aged cell, the cell will no longer function. It will might be alive, just like an old man. They might be alive, but they will not be functional. So what important protein? Tumor suppressor protein. Try to make a short note of tumor suppressor protein P53. This you will be required during your pathology class and also in the medicine class. And those who are opting for oncology, of course, this is one of the important protein which is protecting our basically cell from damage. So they play an important role in the G1 and G2 cycle. What is that? See, this is the DNA synthesis basically occurs during the S phase, that is synthetic phase of the cell cycle. So in the cell cycle, basically there is a G0 phase, and then G1 and S, G2, and this is mitosis is taking place in the M phase. So DNA synthesis takes place during this S phase, during synthesis process, during mitosis. But once it is formed, a cell has been produced, and suppose we are adult, we are undergoing in our body, continuous mitosis is going on, and we are also exposed to environmental insults like X-rays, UV rays, chemical carcinogens, either in the environment or as food we are taking also, or in the industry we are exposed to, we are likely to have damage. So if there is a damage here, it will not allow this stage to go. Again, if it is detected at this stage, so it will not allow the cell to undergo mitosis, it will halt the progression. So there are checkpoints. You see this is G1, G2 checkpoint, this is M checkpoint, this is G1 checkpoint, and there are a number of proteins which are involved that is called cyclins. These cyclins name is because they cyclically increase and decrease with the cell stage. At what stage it is? When the cell is in the synthetic stage, the CDK levels will be high. So nuclear DNA is completely replicated once and only once. This you must remember. So during repair mechanism, I will be telling you that there will be double strand breaks. So during the double strand breaks, during the repair phase, there will be homologous recombination and non-homologous end joining. So homologous recombination occurs basically during the S phase. You remember, and non-homologous joining occurs in the G0 and G1 phase. What are the now cell cycle come. You see there are the 4 phases mainly apart from G0. So G1 phase damage DNA detected, incomplete replication detected in S phase. Then G2 again, if there is a DNA damage, it will not allow the next phase to go. And in M phase, if there is a defect in the spindle, so if there is a defect in the spindle, the chromosomes will not get separated. Then extra chromosome will move to another one cell. That will give chromosomal error that you will read during this chromosomal abnormality diseases. So cell produced because family name, this is a family of protein that cycle during different stages of the cell cycle. So cyclin activate basically cyclin dependent protein kinases. These are called CDK to phosphorylate substrate for progression. What are the? So these are the cyclin and cyclin dependent kinases. What are their roles? That you try to remember. Next. So DNA damage basically, if you look at the present, as I told in the beginning, that we are exposed to mutagenic chemicals, plant toxins, radiations, even oncogenic viruses. Endogenous metabolism, we produce reactive oxygen species, they also damage DNA. And the DNA damage is about 10,000 to 1 lakh molecular lesions per cell per day. But because this constitutes only 0.03%, because we are having 3.2 billion base pair bases DNA. So unrepaired lesions at any particular protein, particularly if it happens with the tumor suppressor protein, it is likely to cause unregulated growth and likely to cause cancer also. So all organisms have evolved elaborate evolutionary conserved mechanisms for its to repair its DNA damage. What are the? This chart you have to remember. There is no way out. So there are different types of mutations. What are the different types of mutation? Whether it is single base, whether it is a 2 base, whether it is a single chain break, or there is cross links. So apurination can be done, or there may be demethylation of cytosine to uracil, demethylation of adenine to hypoxanthine, alkylation of base, and also incorporation of DNA as base analogs. And then these 2 base alterations can be done by UV ray induced thymine or pyrimidine radiation, maybe due to radioactive disintegration, maybe due to bifunctional free radical formation, as I told you, reactive oxygen species, and cross linkage, intra and intra-strand cross linkage, and between the DNA strand and the histone and the proteins. So if there is a cross linkage, then the DNA will not function properly. So these are the basic mechanisms. This is the heart of today's class. We are almost end. So at the base excision repair, remember these 5 points you have to remember at any cost. Base excision repair, nucleotide excision repair, mismatch repair, homologous recombination, non-homologous end joining, and this micro homology mediated end joining and direct repair, these are not that much important because this induces basically mutation and direct repair doesn't occur at all in human being. So all these 5 points you must remember, and the first 3 are required for single strand breakage and last 2 are for double strand damage. What are the? So DNA, this is the DNA structure. Simplified way I have tried to explain the kind of DNA damaging agents, the list I have shown, and this is the DNA damage lesions formed. Basically single strand, double strand, inter-strand cross-link or inter-strand cross-link. This is basically a severe degree of DNA damage. In that condition, this non-homologous end joining or homologous recombination occurs. In other cases, bulky adducts, base excision repair or mismatch repair takes place, and there is a different mechanism altogether. But remember, if the DNA damage is very much extensive, so all the mechanisms might come into the interplay, might come into that side DNA side to make it a stable DNA or as normal DNA as possible. But in that process, usually this repair will not be a perfect one, and some kind of mutations are likely in that situation. So what is the fidelity? Means with what faithfulness it is copying the parent DNA or the damaged DNA or the original DNA is being formed. That is what is called fidelity. You can see the fidelity of non-homologous end joining is only 1 because in a short while you will know what is non-homologous end joining. Homologous recombination is a little bit better, and all these mismatch, base excision or nucleotide excision repair, the fidelity level is quite good. They basically are successful in repairing the damage. What are the? So the diseases which are caused by defects of these procedures or those proteins or enzymes which are required for the repair mechanisms, if they are defective, then these diseases will be produced. You have to remember, because these are common questions in examinations. So, although these cases are shown to you, I am telling you, xeroderma pigmentosa is due to nucleotide excision repair. Try to remember. Homologous recombination, breast cancer susceptibility, ataxia-telangiectasia-like disorder, and non-homologous, basically severe combined immunodeficiency disease. That repair basically do not occur in our system, but it can cause increased cancer risk, neurological abnormality, accelerated aging. What are the? So it is a multi-step mechanism for double strand repair, break and repair. So DNA damage is to be sensed first and foremost, because before whether DNA has been damaged at all, there must be a sensor. So DNA damage sensors are there. These sensors basically proteins. So something has to sense that it is not proper, it is damaged. So sensor must be there. So this is called DNA damage sensor, that transducers and DNA repair mediators. So they will come into the play and they will try to locate where the DNA damage is there and what is the degree of damage. Who will assess? It will not be assessed automatically. Some proteins will be there and they are talking but silent. There is a crosstalk, there is sensor. So cyclins are involved, and ultimately if the repair is complete, then the cell will proceed in the cell cycle, or there will be cell arrest, there will be cell might undergo apoptosis, means programmed cell death, or cell might undergo senescence. That means it will be a stage of dormancy. It will no longer function. It will not undergo mitotic division. So that is called senescence. So basic step in DNA recognition, I think today you are tired. So tomorrow try to come early because I have to cover one more topic. I have to go otherwise fast. So I'll take 5 minutes more only. So basic step in any of the repair mechanism are 4. First and foremost thing is the recognition. Where it is damaged and what is the degree of extent of damage and which method out of these 5 methods needs to be applied in that particular DNA damage segment. Do you understand my point? So identification of recognition of the defect. Second, that part has to be removed first. The damaged part, so damaged part has to be removed. Then once you remove the damaged part, then that has to be synthesized newly. And once you synthesize, then you ligate so that you get back the original things. So these are the 4 basic steps, and for each process, there will be recognition proteins, there will be removal enzymes, there will be synthesis. Synthesis is mainly done by DNA polymerase. As I told you, repair DNA polymerase is because of DNA polymerase beta. Delta, epsilon and alpha also come into the play. That you will see, but the major DNA repair mechanism in the eukaryotes is polymerase beta. And then joining is usually done by DNA ligase, universal. Because a phosphodiester bond is to be formed, and that is done by the DNA ligase. And the cleaving is done by endonuclease. Cleaving because it is within the inside the DNA molecule. So endonuclease will come and it will cut the 2 ends. So these are the basic mechanism. One example I am telling you, glycosylation. This base excision repair, suppose a defective base is detected. Nothing, no more abnormality. It may have undergone perhaps apurination, it has undergone a demethylation. Cytosine has been converted to uracil, or adenine has been converted to hypoxanthine, or there will be alkylation. So what is that? Basically that part only is defective. So unnecessarily the entire thing need not be removed. So that is what is called base excision repair. If you recall correctly, that part, the DNA, the nucleotide part, nucleobase. See that structure of DNA, if you recall. See, there is here is also N, this is also N, to which basically a ribose molecule or deoxyribose molecule. So basically, N glycosidic bond is there, be it purine, be it pyrimidine. So if there is a defect, purine or pyrimidine, this glycosidic bond has to be cleaved because it is attached to, there is no defect in the ribose or deoxyribose. There is no defect in the deoxyribose or in the phosphate. But defect is here only in the purine or pyrimidine. So, and both are attached fortunately with that same N-glycosidic bond. So, a glycosylase enzyme will be required. A glycosylase enzyme will be required to break this bond. So that is the reason why recognition fortunately is done by the glycosylase enzyme. So what is the glycosylase enzyme? Basically recognizes that will cleave this bond and this bond will be destroyed. So it is released. So this structure is bound to the phosphate. So this structure is bound to the phosphate. This is attached to another nucleotide. So this structure is intact. What we have to do, we have to then cleave this phosphate bond. So this endonuclease will also be required. After cleaving by glycosylase enzyme, this sugar and phosphate will remain there, for which you require an endonuclease to cleave this entire unit. Now there are 2 types of glycosylase. One is having only glycosylation activities, that is called monofunctional, and the second one is called bifunctional. So bifunctional glycolase, this is I have told you, monofunctional glycosylase is having only glycosylase activity, but bifunctional glycosylase also possess AP lyase activity. Remember what is the difference between lyase and hydrolase? Lyase also cleave bond, but they don't require water. Hydrolase cleave bond, but they require bond. So if they are having an AP lyase, once this part is removed, what is done? This is basically a AP site is formed. AP site means, or A basic site is formed. AP may apurinic or apyrimidinic site will be formed once this structure is removed. That AP site is formed. AP site means A basic site or apurinic or apyrimidinic site. So if there is a AP lyase activity is there, so a bifunctional glycosylase will not require any AP endonuclease activity. So that is the one part I am telling you. Now here you can see there is a small single nucleotide defect. So then the short patch repair pathway will be used. That will basically clip that part, and the new DNA, new base will be synthesized by the DNA polymerase, and the two ends will be ligated by DNA ligase. But if it is a part of if it is more than one base defect, so base excision requires 2 or 10 base up to, that is called a long patch repair. Here that stretch of DNA will be produced. You see here there is no flap, but here 2 to 10 is there, then there will be a flap will be there. So that flap will be removed and a new strand will be formed. Then the new strand will be flap endonuclease will be there. It will remove the defective flap. This flap is defective because 2 to 10 nucleotides are defective. That is the reason a flap is created. So flap endonuclease will clip that flap, and then the rest of the proteins, these nucleotides will be synthesized, and they will be joined by DNA ligase. So whether you require a single nucleotide replacement or 2 to 10, whether one short patch repair or long patch repair, that will be decided, because that's why I told you that sensor must sense what type of defect it is, whether should I go for base excision repair or whether should I go for nucleotide excision repair, or if there is a mismatch, then mismatch repair, if something more, cross linkage or there is strand breaks, then other methods. So with that I conclude. Rest of the mechanisms tomorrow I will discuss. Yes. That out of so many bases, only one base is by chance methylated or had undergone apyrination or apyrimidation, whatever it is. So only one base is involved, then the short patch will. Suppose if the damage is a little bit extensive, so more than one base, up to say 10 base is defective, then the base excision repair will involve the long patch. If it is something more than nucleotide excision repair will be there, that I will discuss tomorrow. So this is basically, you understand. Here, if this is a nick will be made here. Polymer যেখানে লাগবে, এই polymer যেখানে লাগবে, polymer যেখানে লাগবে, DNA লাগে যেখানে লাগবে, endonuclease লাগবে, endonuclease লাগবে না, endonuclease টা ভরে লাগে, polymer endonuclease লাগবে, এই lyase activity দেওয়া আছে, এই যে lyase দিয়েছি, if the lyase activity আছে, lyase is incorporated within the, I told you, if the lyase activity is there, then there is no need for endonuclease activity. If glycosylation is n glycosylate bond এখানে রয়েছে, n glycosylate only purine defected হয়েছে, suppose এটা cytosine ছিল, এটা uracil হয়ে গেছে, and this DNA damage sensor has sensed that it is not cytosine is uracil, or it is not adenine, it is hypoxanthine. So this is to be removed. So for that, glycosylase enzyme will be required. You understand my point? After that thing, this will be removed, then these two parts will be there. So the entire unit has to be removed. So basically, then it will cut here by endonuclease, or if it is by a lyase, already built-in lyase, AP lyase is there. So that lyase will cleave here without requirement of any water. Endonuclease will cut, it will call a hydrolysis, it will require water, that's it. বোঝা গেল ব্যাপারটা? সেকেন্ডটা দেখো, একই জিনিস আছে। এই দেখো ডিএনএ পলিমারেজ ডেল্টা আর এপসিলন, এই রিপেয়ারটা করছে ডেল্টা এপসিলন করছে, ওটা ইয়ে করছে ডিএনএ বিটা করছে না, কিন্তু এটা লং প্যাচ আছে। বোঝা গেল ব্যাপারটা? আগেটা দেখো, এই দেখো ডিএনএ পলিমারেজ বিটা করছে এটা। বোঝা গেল ডিফারেন্স? নাও ইউ আন্ডারস্ট্যান্ড দ্য ডিফারেন্স। If you read a little bit, if you go through everything is written here very clearly. So DNA polymerase beta is involved in this process, single base. In contrast, DNA polymerase new strand, if such things are there, DNA polymerase delta and epsilon is functional. So because leading strand, lagging strand synthesis is there, you read it properly, so you will get whether it is leading strand or lagging strand, but here there is no leading strand and lagging strand. It is simply a mere repair. That's why that part was not written in that book so exclusively. But if you read a little bit, if you do a scientific study, you will definitely get other functions of D and E and other units also. Okay, with this we conclude. Try to maintain a decorum. Don't irritate me. I'll take one more maybe tomorrow's class, and that's end of your batch. I am not in a mood to take also a class. Okay. Question you see, a 5-year-old boy presented with blistering photosensitive lesions, diagnosed as xeroderma pigmentosa. What is the molecular basis of this disorder? Write a notes on different agents of DNA damage. You people kept on gossiping. Enumerate 6 steps of DNA damage repair methods, which I have told. I have told the different notes on different agents of DNA damage. Name the prokaryotic DNA polymerase involved in DNA repair. I have told earlier also. Name the eukaryotic DNA damage with the roles it was discussed in the first line. And none of the students could answer. Hardly maybe a few. But this was not a complete answer. You are talking too much, na? The day you will get a question paper, in your final examination, you will have the charm. And I expect a still difficult question. Because the day every year I am seeing some amount of difficult questions are being set. This is indeed a difficult question. I definitely say, xeroderma pigmentosa you are not supposed to know at this stage. But the first question, so much demotivating, I am telling you. I am criticizing rather that this question ought not to be given to a first year student. But this basically is, this is I am trying to describe because I knew this question was there. So therefore I am trying to give you in detail whatever I could give, so that you perform better. It is my wish that all of you pass in this year also. Unlike that batch, don't fill in that numbers. That will be demoralizing to you, demoralizing to us also. Now can you answer to these questions if you do not read it properly? And the marks are also given very clearly. Unnecessary writing will attract 0 marks. Very specific points. Chalo.